MRI.
Thank you for the introduction. So the aim of this work is to open a new window onto brain tissue microstructure by developing a method to quantify cell body or soma non-invasively using diffusion-weighted MRI. So the current conjecture in microstructure imaging model the cellular compartment in the brain as you see in this picture. So the diffusion of water in the cells are modeled like diffusion in cylinders, more or less randomly oriented. And as you can see, the soma is not taken into account, or rather, the way the modeling works does not allow a direct modeling of the soma. In fact, the standard model at the moment is comprised of two compartments: intraneurite compartment, diffusion of water molecule inside the neurites, and then the extraneurite compartment, which eventually include the soma contribution. Now, this model has been very successful to describe white matter, but as you may understand, to describe gray matter may not be sufficient. In particular, if we look at this data, these are directional averaged signal as a function of b value, okay, 1 over square root of b. You can see that in white matter, you have a signal very different from gray matter, in particular at the high b value region. And here, you can notice there is a specific curvature of the signal. Now, this curvature is something which is typical of gray matter, and it's much less pronounced in white matter. Current models cannot explain this curvature. So what our hypothesis, what we propose, is that that curvature is a signature of cell bodies or soma. So if we can model that curvature, we can quantify the soma. And we propose here a model to do this. Our model is a compartmental model, but this time we characterize the intracellular space, subdivided in two subcompartments, intraneurite and intrasoma. And then the extracellular space now doesn't contain the soma contribution. So we aim to model directly the soma. I will skip the mathematical details of the model because there is nothing special here, but it's a three-compartment model. You have the intraneurite, intrasoma, and extracellular. Our model relies on some assumption because, of course, if you want to have your system described by three separate compartments, you need to do some assumption. The first assumption is that soma and neurite can be modeled as two different compartments that don't exchange water between them. The second one is the intracellular compartment and the extracellular don't exchange water. And then we fix the diffusivity of the soma. So while the two assumption B and C has been validated under a specific experimental condition, the assumption A is not obviously valid. But as you will see, we will show with numerical simulations that in some experimental condition that holds. I would like to spend some words on the model parameter because I think it's important by model design that these model parameters can be linked to some interesting features of the brain cyto and myelo architecture. In particular, we will aim to measure soma size and soma density as an indirect measure of the signal fraction from the soma compartment and the size of this compartment, and the intraneurite density. So the roadmap for this model, as I said, first of all needs to validate the assumption A. And we do that with advanced numerical simulation of brain cells. If you are more interested about this, I have a poster tomorrow. Then we will demonstrate in vivo in human using the MGH-HCP dataset that the contrast that we get is actually meaningful. And then we started a validation ex vivo in mouse to actually compare our soma density maps with, for instance, the API staining. So first of all, numerical simulations. As I say, that this is one of the important assumptions, that if we have a cells where water can diffuse in neurites, go into the soma, and jump in another neurite, well, we characterize it like water only in the neurites and water only on the soma. This is not obvious that it should work. So what we did, we simulated two conditions. One, we allow the water to go between this compartment, and the other, I stop it at the boundary. So whatever it is in the neurite stays in neurite, whatever it is in the soma stays in the soma. And we compute the apparent diffusion coefficient in these two conditions. The difference between these two diffusion coefficients, if it is small, it means my model is a good approximation. And what I mean for small, well, in this case, we put a threshold at 10%, taking into account the possible noise in the experiments. And here you see the results for four different sizes of the cellular compartment that could represent a large neuron and a small microglial cell, for instance. And what we found is that if you work at diffusion time lower than 20 milliseconds in your experiment, then the model assumption is correct and the model works. And here you can see in color the lines of the simulated signal, and in black dashed line our model. It perfectly matches the signal if we are below 20 milliseconds. If we go above, the model starts failing. So we can design an experiment in this sweet spot and try to measure the soma. Luckily, we didn't need that, because on the MGH dataset, the adult diffusion dataset, the sequence parameters are almost ideal for this experiment. Diffusion time lower than 20 milliseconds and quite high p-value. So what we did retrospectively analyze the 25 subjects from this dataset with this preprocessing, and we used also FreeSurfer to extract the cortical surface and then maps our parameters, model parameter, onto the cortical surface. So here is an example for one subject of the maps you can obtain. And in particular, I would like to point your attention on the contrast of the neurite signal fraction and the soma signal fraction, which remarkably mirror myelin distribution in the brain and the cytoarchitecture of the gray matter. Then we have other parameters like, for instance, the size of this soma compartment and the diffusivities. To convince you that the contrast is actually meaningful, I compare here just qualitatively the signal fraction from the soma compartment and the Nissl staining. And you can see how this contrast mirrors very well the Nissl staining of cell bodies. And on the other hand, the signal fraction from neurite mirror very well the myelin staining. To be a little bit more quantitative, we performed the average over 25 subjects, and we projected the soma signal fraction onto this surface. And here you can see Brodmann parcellation overlaid on this signal fraction density. The boundary between our gradient in the value of signal fraction and the boundaries of Brodmann areas are very well matched. They match very well, which is quite interesting. Even more, if we look at specific region known to be classified by Brodmann based on density of the cell body and also arrangement. But in this case, you can see how our contrasts mirror very well. Where we expect high density, we measure high value. Where we expect low density, low value, even in an interesting region like Brodmann area 44 and 45. And this contrast is, here I show you just an example of different subjects to show you that the contrast makes sense also, not only on average. As a validation, we performed the experiment in the same regime, and we did the DAPI staining for cell bodies. These are the maps for the mouse, and you can see a nice layering in the soma signal fraction, hippocampus, cortex, olfactory bulb, and cerebellum. Size-wise, it also makes sense in the cerebellum, where you have a higher density of granule cells. The dominating size we measure is small, while in the hippocampus you can distinguish nicely or the arrangement of the big pyramidal cells. And this is the neurite fraction. Direct comparison with histology from the same brain show that the contrast is actually mirrors very well the layering of the cortex. Here you see on left the maps we obtained with MRI, and on the right the histology on the same brain. And here I scale the histology to match more or less the contrast in MRI. To be more quantitative, we measure the correlation between soma signal fraction that we measure and the stable staining, and you can see there is a nice strong correlation, linear correlation between the two. So to conclude, this work introduced and start a validation of a new method for non-invasive soma imaging for the first time. Also, it's a new model that allows to quantify at the same time both cyto and myelo architecture, and it provides a new set of biomarkers of potential value for biomedical applications and pure neuroscience. We are still performing the validation properly with ex vivo experiment, but I think some of the results are quite convincing. I would like to thank you all for your attention and all my collaborators. Thanks.